The objective for Lab 1, which is parasitology, firstly to determine the kinds of parasite found in the fish sample. Secondly, to discover any clinical indication produced by parasites in the fish. Thirdly, to identify the parasite kinds prevalent in a specific fish species collected from various body sections.
For introduction, parasitology studies parasites which live in or on a host and derive nutrients from it. They can cause various diseases and can be found in plants, animals, and humans. There are three main types, which are protozoa, AMIN, and arthropod. Parasites can infect humans through injection, contact, inhalation, and vector-borne transmission. Injection occurs when people eat or drink contaminated food or water. Contact occurs when people touch contaminated soil or water. Inhalation occurs when people breathe in contaminated air, and vector-borne transmission occurs when people are bitten by an infected insect or arthropod.
This slide shows the material that were used in this process.
Next is the methodology. The fish was killed first, then placed in a density pan to observe a parasite on the external fish. After that, the gills were carefully removed by severing the blood vessel, and the gills were placed into the slide. After that, the mucus on the gill were smeared in the slide. The parasites that were found were examined using a microscope. Lastly, the process was repeated using the sardine brand ads.
When Lab 1 was conducted, the specimen of ikan krisi did not show the presence of parasite on the outside of its body during the observation process, while inside its body there were parasites in the gill, which is Dactylogyrus sp, which can impact on the fish health. So they attach to the gill tissue, which causing damage and affecting respiratory function. So for the life cycle of Dactylogyrus, typically it has a direct life cycle, which with both larvae and adult stages that can be occur on the gill. So the egg hatch into larvae, which then attach to the gill surface, develop into adult and reproduce. So proper identification and understanding their life cycle are crucial for effective parasitology management in aquaculture for the future.
In conclusion, threadfin bean and sardine species have distinct parasite, which the threadfin bean finding a monogeneous parasite, Dactylogyrus, in the gills, while the sardine had parasite absent. It is because canned sardine typically undergo high heat processing during canning, which effectively kills any harmful parasite that might be present, and also because we examine only a small number of sardine. Therefore, consuming commercially canned sardine is generally safe from parasite infections.